Hello everyone, welcome to class 9, Science, Chapter 2, Cell, the building block of life. So today we are going to see the book back exercises. So what we saw in this chapter, children, we studied about the cell. We know that cell is the structural and a functional unit of all living organisms. We studied about a prokaryotic cell, eukaryotic cells, and its different organelles present in it. And also it is having a cell membrane, and plants have the cell wall. Yeah. And also we studied about different organelles, yes, and the functions of the organelles. And also we studied about plastids, different types of plastids, and its functionality and cell division. Let's start with the question. So the first question, differentiate between the following pairs based on the clues given in parentheses. So cell membrane and cell wall. So based on its permeability, and rough endoplasmic reticulum and smooth endoplasmic reticulum based on structure. Chloroplast and chromoplast based on pigments. Yeah, let's see the answer. So the cell membrane, they are selectively permeable. So it can control whatever enters and leaves. And cell wall, it is fully permeable. So everything can pass in and out. When we talk about the rough endoplasmic reticulum, it is having the ribosome on its surface. That is why it is giving it a rough appearance, and also it makes proteins. When we talk about smooth endoplasmic reticulum, it is having it don't have ribosomes. So the surface is smooth, and also it makes lipids or hormones. Chloroplast. So chloroplast, the chloroplast, they are having a chlorophyll, which is a green color pigment, which helps in the process of photosynthesis. Chromoplast is, they actually give color to the flowers and fruits. So what are those colors? Yellow, orange and red colors. Next is, two similar animal cells are placed in two different solutions. Yes. So two similar animal cells are placed in two different solutions. So cell X and cell Y. So the cell X is placed in pure water and cell Y is placed in a concentrated salt solution. Cell X swells and cell Y shrinks. So here they have given some statement. They are asking which statement is explaining this one. First, let us see. Salt molecules moved into the cell. We have already studied this chapter, right? Will salt molecule move in? No, only water molecule. So the first one is wrong. So water moved into cell X, and more water moved out of cell Y than salt entered. Now this is also wrong. Water moved into cell X and out of cell Y through the cell membrane. Yes, this is correct. And solute movement caused osmosis in both cells. Solute will move, children? No. So this is the answer. So water moved into cell X and water moved out of cell Y through the cell membrane by a process called as osmosis. The cell membrane is selectively permeable. So water only pass, but salt, it won't allow the salt to pass through. That is why in cell X, water moves in, and in cell Y, water moves out because it is placed in a concentrated solution. Identify and match the cell parts. So here we are given the diagram. So A, B, C, D, yes, D, E, F, G, and also they have given functions to match. First one we have to study this one, and then we have to match with this A, B, C, D. So let's take the first one. Yes. So mitochondria. So where is A, children? Can you be able to see? So this A is nothing but mitochondria. What is mitochondria? Powerhouse of the cell. Then what is this thing? Can you be able to spot where it is? Yeah, it is a site of cellular respiration. So this is A. Yes, this is A. This is A. And then next one is B. B is nucleus. So what it does, it controls the cell activities. It is like a manager of the cell. So B controls the cell activity. So this is the B. Then what is the C1, C is Golgi apparatus. So this is C, this is Golgi apparatus, right? This one. So can you see the functionality over here? Can you search? Yes, of course. See, it packs materials from ER. What it does, it packs materials, packs and stores materials received from ER. So it stores the material, it modifies and packs it. Yeah, shipping, everything done by the Golgi. So for which one? For this one, this is the C option. Sixth one is C option. And D. D, can you able to spot? This is the, can you able to see the stroma? Yes, the thylakoids, everything. So this is the chloroplast. So chloroplast, photosynthesis. So where is photosynthesis? Can you spot out here? Where is photosynthesis? Is food production. Can you able to see? Yes, helps in manufacturing food. So this is a helps in manufacturing food. This one. Then E. What is E here? It is the cell membrane. So it is showing the outermost membrane, cell membrane. So what cell membrane? It separates the cell from the surrounding region, right? Yeah. So where it is? Can you able to spot? Yeah. Separate cell contents from surrounding. Separates cell contents from surrounding. So this is E. And then F. F is the cell wall. F is the cell wall. So what cell wall does? It gives rigid nature to the cell. So where it is, can you be able to spot? Yeah, it's here. Fifth one. Provides structural rigidity to the cell. So this is F. And then G. G is here. This is a big one which is present in the plant cell. What is this? Vacuole. So what is the function of vacuole? It's gives storage. So we can store starch, yes, oil, waste, everything will be stored in the vacuole. Where is this one? So this one is a remaining one. Storage organelle that also provides rigidity. So this is the answer. So G. Okay, children, let's move on to the next one. So next we are going to see which option correctly places organelles under the given categories. So options I have given plant cells and animal cells also we can. So we have to find out the correct one. When you take leucoplast and cell wall, where it is present, cell wall in animal cells we don't have cell wall, only cell membrane is present in animal cell. But this leucoplast and cell wall is present only in plant cells. So first option is wrong. Next mitochondria and ribosome. Let us compare this with the answer also children. When we take this mitochondria and ribosome, it is present both in plant as well as the animal cell. Okay. So this option also we cannot take, it is wrong. Third option when we take a cell wall. Cell wall is present, is correct. It is only present in plant cells. And Golgi apparatus is present in both the plant as well as the animal cell. When we take the lysosome and endoplasmic reticulum, these both are present in plant as well as the animal cell. So our correct option is option 3. So cell wall is present in plant cells but absent in animal cells. The Golgi apparatus is found in both cell types. So why other options are wrong children? Because when we take the first option, both leucoplast and cell wall are present only in plants. So it is absent in animal cells. So applies to both, making the option incomplete. Right? When you take the mitochondria and ribosomes, they exist in both plant as well as animal cells. When you take lysosome and endoplasmic reticulum also, they are present in both plant as well as the animal cell. So next question is based on plastids in plant roots. Two students, Renu and Rohit, discussed plastids. Renu said all parts of plants, even roots, contain plastids. Rohit disagreed, saying plastids are absent in roots since both roots are underground and don't photosynthesize. So we have to find who is correct and justify your answer. Actually, all living plant cells contain plastids. Plastids are only present in plants, right? So, but the type of the plastid varies by tissue. When you take the roots into account, it is having the leucoplast, which is a colorless plastid which stores the starch, oils, and proteins. Green leaves, they have big green pigment, you right, because of the chloroplast, which helps in the photosynthesis process. And flowers and fruits coloration is because of a chromoplast. It is having the colored pigments. So Rohit, he has mistaken, he's confused plastids with chloroplasts. But plastids are not only chloroplast, it is a chromoplast and leucoplast also. So next question based on cell organelle. Mitochondria and chloroplasts are two important organelles in a plant cell. Discuss how these two organelles are structurally similar to each other and functionally different from each other. Let's see the answer. So structural similarity, if you see, both are double membrane bounded. They contain their own DNA and ribosomes, and they have inner membrane folded for surface area, right? So cristae in mitochondria and thylakoid stacks in chloroplast. So when you see the functional difference, mitochondria release energy by breaking down glucose, the cellular respiration. It gives out ATP, right? So it is found in plant as well as animal cell. Chloroplasts make food using sunlight, photosynthesis, found only in plant cells. So which pair contains DNA? Which of the following pairs of cell organelles contain DNA? Chloroplasts, ribosomes, mitochondria, nucleus, Golgi bodies, ribosomes, nucleus, lysosomes. Let's see the answer. Second option, mitochondria and nucleus is the correct one, because see, both organelles contain DNA. We know our DNA, our chromosome, everything, it is present in the nucleus. And mitochondria, they have their own small circular DNA. Why others are wrong, children? Because when you take this ribosomes, it is having RNA and protein. And this Golgi bodies and lysosomes do not contain DNA. And chloroplasts, if we take, they contain a small DNA, right? But option 1, that is pairs them with ribosomes which don't. So it's wrong. Okay. So next is an carrot experiment. A researcher placed one carrot in plain water, cup A, and another in concentrated salt solution, cup B. After 24 hours, she recorded her observations. What hypothesis does she want to test? What hypothesis is this? And what would you suggest for the improvement of this experiment? What we can suggest? And then why does the carrot in plain water stay stiff but in salt becomes limp? For this answer is, first answer is osmosis. So what happens in osmosis? Water moves out of plant cells when placed in a high salt, that is the hypertonic solution. So this process is osmosis. What improvement we can suggest? We can weigh both the carrots before and after in order to see the difference in weight, and also repeat with the multiple carrots, and also vary salt concentration, and we can control the temperature also. Depends upon the temperature also, the experimental result varies. And next question, why does the carrot in plain water stay stiff but in salt become limp? Because in plain water, water enters the carrot cells because of the turgor pressure and it becomes stiff. But in salt water, what happens? Water leaves the cells. Yes. So what happens? That is why it is undergoing plasmolysis. So what happens during plasmolysis? The cell membrane just move away from the cell wall, and that is why it is becoming limp and rubbery. Here we are going to indicate the presence and absence. So indicate the presence or absence of these structures in bacterial as well as the animal cell. So let us see. Let us see the answer for this one. When we talk about this chromosome, see, only we have nucleoid in a bacterial cell. Yes. So in animal cell we have chromosomes. When we talk about bacterial cell, nucleus is absent, mitochondria is absent, Golgi complex is absent, and chromoplasts, they are absent. But all these things, everything, they are present in plant cell except chromoplast. Why chromoplasts? They are plastids. They are present only in plant cells. Next, see the 4 potato egg cups experiment. 4 peeled potato halves scooped into cups. Cup D is made from a boiled potato, all placed in a beaker of water. Empty cup, let us take A, empty cup as the control. And in B, we are taking 1 teaspoon sugar in cup. And in C, we are taking 1 teaspoon salt in cup. And in D, we are taking 1 teaspoon sugar in boiled potato cup. So, questions. Why does water gather in B and C? And second question, why is A necessary? Next, third one, why does not water gather in A and D? Let's see the answer. So when we talk about the B and C cup, so it makes the inside thing as hypertonic. So when it becomes hypertonic, what happens? More solute than water around. That is why water moves in by osmosis through the potato cell membrane and water gathers in the hollow cup. But what happens in cup A? Cup A acts as a control. So no solute. So we can prove water moves only when a solute gradient exists. When you take the third question, why does not water gather in A and D? A, no solute, so no osmosis. D, boiling destroys cell membrane. So without intact membranes, osmosis cannot occur. So incorrectly matched pairs, we have to find out. Identify the pair that incorrectly matches the cell organelles with this function. First one is ribosome protein synthesis, then smooth endoplasmic reticulum, lipid and cellulose synthesis, lysosome digestion of foreign agents. Let's see the answer for this one. Second option is incorrect. Why? Because when we talk about smooth endoplasmic reticulum, synthesizes only lipids and hormones. It don't synthesize cellulose, because where we find the cellulose? Cellulose, which makes up the plant cell wall rigid, right? So it is produced by enzymes located on the cell membrane, not by the smooth endoplasmic reticulum. Why others are correct, children? Why? Because ribosomes are the site of protein synthesis. So first one is correct. And third one, lysosome contain digestive enzyme which breaks the foreign particles and bone out organelles. That is why it is also correct. So the incorrect one is the second option. What outcome do we expect if all the mitochondria are removed from a eukaryotic cell? We cannot think a life without mitochondria. Let's see why. So the cell cannot survive long. So what will happen? Cellular respiration cannot take place, right? So glucose breakdown cannot happen and no ATP will be produced. And the energy reading activities like your active transport, protein synthesis, muscle contraction, cell division, everything will stop. And cells should rely only on anaerobic respiration, glycolysis, where what happens, only 2 ATP molecules are produced. But when we talk about this mitochondria, 36 ATPs are produced. We cannot compare the 2 ATP and 36 ATP. So eventually what will happen, after some time the cell could die due to severe energy shortage. That is why we are calling mitochondria as the powerhouse of the cell. So what inhibits tumor formation? So which phenomenon inhibits the formation of tumors in the human body? Can plants also develop tumors? Explain. So let's see the answer. So contact inhibition is the phenomenon. So what happens here? So contact inhibition means it stops the normal animal cells from dividing once they touch the neighboring cells. So this keeps tissue organized and it prevents uncontrolled growth. So what happens? The cancer and all means what will happen? It will be like uncontrolled growth. So when does this cancer happen, children? So the cells, they lose that contact inhibition, they keep dividing uncontrollably and they form tumors. When we talk about the plants and tumors, plants actually they do not show this contact inhibition because they have a rigid cell wall. But because of some bacterial disease, bacteria caused by bacteria, they show tumor-like growth called galls. So we know the disease as the crown gall disease. Yes. So what happens? They are not cancers actually, but they are uncontrolled cell proliferation, or we can say cell division. The cell membrane is made up of proteins and lipids. Which cell organelles help in the synthesis of the cell membrane? Trace the path of these compounds from synthesis to the cell membrane. Let's see the answer for this one. So ribosomes. So what happens? See, these ribosomes are seen on the rough endoplasmic reticulum. Yes, and they make the membrane protein. And smooth endoplasmic reticulum makes the lipids. So both are packed and they are transported by means of vesicles and they are sent to the Golgi apparatus. What Golgi does? It receives it, it modifies it, it packages it, and it sends in the form of new vesicles to the cell membrane. So what the cell membrane does, these vesicles fuse with the membrane and they add more new lipids and proteins to it. Yes. So what would happen if gametes are formed by mitotic division instead of meiosis? We already know in mitosis, yes, what happens in meiosis. So mitosis happens in somatic cells or the body cells. Meiosis happens in reproductive cells. Let's see the answer. So each gamete would carry the full chromosome number of the parent instead of half. So in the mitosis, yes, two identical data cells will be having, so it will be having the full chromosome number. But what happens in meiosis, half the number of the chromosomes only will be there. Yes. So because of this consequence, what happens after fertilization, the offsprings would have double the chromosome. Actually, how many pairs of chromosomes? 23 pairs of chromosomes will be there. But instead of that, it will have, see, you can able to see, I have given you examples also. Instead of 46 in humans, 92 will be there. So this doubling could cause some chromosomal disorders or genetic disorders or developmental abnormalities or non-viable offsprings. Even sometime early miscarriages also may occur. Yes, and no genetic diversity. Yes, and meiosis halves the chromosomes specifically, so fertilization restores the normal number. That's why it is essential for the sexual reproduction. So case study. Farmer Deepa harvested amla and lemons. She turned excess into pickles, murabas, and sharbat by adding salt, sugar, or jaggery, preserving them in glass bottles and shifting from farming to agro processing. So, what are the questions? Scientific concept used. Yes. How does salt or sugar prevent spoilage? Suggest a healthy recipe and a scientific value. Let's see the answer. So concept is osmosis. So water moves from a low solute to high solute. So mechanism, when the high salt or sugar so makes the surrounding hypertonic. Because of this, water happens. Water is drawn out of the microbes. It can be a bacterial cell or a fungal cell. So microbes die or they stop multiplying, and it also preserves the food. So food don't spoil, right? And then healthy recipe like lemon honey salt sharbat we can give, or amla murabba, or sun dried tomatoes with the rock salt. Yeah, these and all we used to have now children papada. Yes, so many things we have, and they are natural preservation without chemicals. Scientific values. So what scientific values we can give? We can actually scientific thinking, sustainability, that is a main thing, and food security, and also reducing post-harvest waste, and woman empowerment and traditional knowledge. These are some scientific values. So the journey beyond projects and reflection. Okay, so what we can do? The journey beyond hands-on projects. So we can use some software or digital tools to create animations or simulations of cell divisions. So you can share with your class, or you can build a model of the synthetic cell using low-cost, eco-friendly material. Yes, so many things are available at a home. Yes, are very eco-friendly things. You can create a model, cell model, and build a mitosis or meiosis model with classmates for your science exhibition in your school. As team, you can reflect the teamwork over there, or you can develop a Nukkad Natak, a street play for community awareness about the functions of cell organelles. So the quest continues. What is the future of developing synthetic cells using non-living chemicals? If a synthetic cell is developed, what may be the related ethical issues? So these things are, yeah, you can have a debate in your class also. Yes. So what we saw, this the chapter wrap up. Yeah, what we saw, you have to revise everything, read at a glance. Yes, and you focus on areas where you struggle, and then reflect. So what you have learned and relate with the everyday life, like a preservation techniques you can use in pickles. And when you see a plant, you can say which part is having what kind of plastids you can, and also how you are healing wounds. Yes, everything you can relate it and then refine. You have to practice the diagrams, which is a very important thing, children. And you can compare different things, whatever you have studied, and also case study questions to sharpen your skill. So keep in touch, children. We have completed this chapter. Yeah. Wait for the next upcoming chapters. Yes. Thank you.